Welcome to Faculty Focus, the podcast of the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences of the University of Antwerp, live from our annual Faculty Research Day, where we highlight the research of our professors and PhD researchers. I am Teun.
And I am Marleen.
And our next guest on the podcast is researcher Rita de Sousa. Welcome to the podcast, Rita.
Thank you.
She works at the Department of Veterinary Sciences at the Gamete Research Centre on the production of follicles from oogonial stem cells and calls this the long-awaited solution for fertility preservation and restoration. So, in the introduction to your research, you state that more than 80 studies challenge the dogma that females are born with a limited number of oocytes and that ovaries lose their capacity for oocyte renewal at the time of birth. Is this the result of a male-dominated science landscape, you think?
Okay, starting very controversially, I think.
It was your introduction, not mine.
Yes. I wouldn't. Yeah, the scientific world was indeed male-dominated, but I think there's also female scientists that keep on pushing that this dogma is strict and fixed, and it will never change. And I think that's not a way to approach science. You should always be open-minded to discover new things. And that's why my research always keeps on fighting and fighting and fighting.
So it's a bit like the Earth is flat and the Earth is round type of question, right?
Yeah.
So is the long-held belief that women are born with a fixed number of egg cells now really being challenged?
Yes. Ever since 2004, there have been so many research studies challenging this dogma, and there has been offspring produced in mice already from my oogonial stem cells. So, yeah, you keep on giving evidence that this should and can be challenged.
So what could this mean for the infertility treatments?
Yeah, it means a completely change of way of thinking. Anyone that, well, imagine if you're a menopausal woman, you never had the chance to have your own kids, and now you want it for any other reason. Then the oogonial stem cells brings a way that you can't do it like that. Or, for example, if you are a prepubertal girl and you had cancer or something, and your oocytes were compromised by it, you don't have oocytes anymore, then that's a way that you can still be a woman and have your own kids. If men can have spermatogonial stem cells and have kids until they are 90 or more, women should be able to also. So, yeah.
And maybe you have to define the oogonial stem cells for us.
Yeah, so it's a term for germ stem cells that are found in the ovaries, and they are already there. So a stem cell should and can be differentiated into something else. And these oogonial stem cells, they have the ability to differentiate into new oocytes.
Okay, so. And you talk about pre-existing oogonial stem cells. What can you elaborate on that?
So pre-existing means that they are already there. So I'm trying to say that the oogonial stem cells already exist in the ovaries from the day we are born. That's different from the induced pluripotent stem cells, which is something we create in a lab.
Okay. And how do you identify these cells?
Yeah, it's with a technique called fluorescent activated cell sorting, where we sort for a very specific germ cell marker called DDX4. And then the oogonial stem cells, they are extracellular positive for this marker, whereas all the other germ cells that are in the ovary will not be stained for this. Okay. And then we can really separate our population from everything else that's in the ovaries.
Okay.
Do you know where in the ovary it's resided?
Really. We still, that's one of the controversial topics where they actually are. Some people say they are more in the ovarium epithelium surface, but they can be anywhere in the ovary. That's something that we still need to discuss and research.
You also use ovine 3D organoids.
Yeah.
What are those exactly?
Yeah, so it's a very fancy way of saying I couple cells together and I form like a kind of 3D structure, like a ball with a mix of cells. And I use my oogonial stem cells and other type of somatic cells that are already present in the ovary, and I bring them together, and then I make kind of a ball of cells.
And do they just form a ball together, or do you have to add other factors
no,
for
we
it?
use a centrifuge, and then the cells couple together, and then they are in specific plates, and then they just make a ball.
Okay, make it sound so easy.
Yeah.
And how big is that ball? Do you have an idea?
It's really, really small.
So nothing olive-like, it's just more...
Yeah.
It's smaller than that.
Yes, it's very small. You can see it in the microscope clearly, but not
not with the naked eye.
Yeah.
And your research, or the research around this, started in mice, but you focus on large animal models.
Yes.
Yeah. Why?
Yeah, we started with a pig to bring it closer to human research. So pig is very more similar to humans than mice is. And there's a very less scientific way to say that everything works in mice. So we wanted to prove that we can show it in larger animal models. And we also wanted to look not just into human research, but also endangered species research. So we wanted to be as broad as possible. And with a larger animal model, you can bring the research closer to these kind of human and endangered species. But we also we do pig, we do cow, we do sheep. So as many as possible, basically.
The llamas here as well?
No, not the llamas. Yet.
So is this an invitation?
Yeah, well, the thing is, we always need to wait for an animal to die. So for the pig and the cow, we get it from the slaughterhouse and also the sheep. But if an animal dies at the zoo or something, there's always a possibility that maybe we can get an ovary and then we can do some tests.
Okay.
But it's a matter of waiting.
Because on the podcast, we already had Professor Ruud Appeltand.
Yes, she's my promoter.
Yeah, this is your promoter. And how does your research fit in her work with in vitro gametogenesis? Yeah,
so basically what I do is in vitro gametogenesis, but her real true passion is indeed about the rhinoceros. Yeah. And so pig is a bit far away still from the rhinoceros, but we do have tapir ovaries that maybe eventually I can also use and get oogonial stem cells and then produce new follicles, and then she will be very excited.
Okay.
Is there something we didn't ask you about your research and the talk you will give during the research day?
No, I just hope everyone likes the talk and that it's clear enough to understand. It's a very new topic, and I hope everyone can be open-minded about my work.
Okay. And maybe we want to finish off with a broader question. What advice would you give to a starting PhD researcher?
Yeah, be realistic. You need to have a lot of passion for the work you're doing, more than any kind of normal job, because you will struggle. That's the reality. You always imagine, it's going to get great. In my case, I would produce like a mini pig from the oogonial stem cells. It will be amazing. But that's never going to happen. That's not going to happen while I'm probably alive. Yeah. So you need to tone it down, have a passion, and aim for like the world, but be realistic that it will probably not gonna succeed.
Okay.
Mm-hmm. And
just take it step by step.
Yeah, your research is just a piece of the puzzle.
Yes,
right? Yeah.
indeed.
And the whole puzzle is difficult to achieve in one PhD.
Yeah.
That's true.
Okay. That's good advice, I think. Thank you very much, and good luck with your presentation this afternoon.
Thank you.
Thank you.